Good morning, good afternoon, good evening, dear children. We are coming to the fourth module under genetics. See, living organisms have a life cycle. When they are born, they grow, they develop, and then they die. The same way, any cell which is born after a cell division goes through a life cycle which is called cell cycle. It will grow, it will develop, it will perform certain functions, and then it will perish. So, in this module, basically, it is all about cell cycle. Apart from that, there are a few terminology related to human chromosomes which we will need in the upcoming modules. Those terms are karyotype, diploid cell, a haploid cell, autosomes, and allosomes. So we will be dealing with those basic definitions also in this module so that the next modules will be comfortable for you. But before we proceed, as usual, a revision of the previous module through a PJ. Yes, you heard it right, poor joke. If you think a teacher is incapable of that, check out. So, cell cycle refers to all those changes which happen in a small tiny cell which grows, develops, and then till it perishes. Now, which are the things happening in the cell? The first will be the growth of the cell. This growth will be facilitated only if it synthesizes more proteins, more amino acid, more nutrients so that it can grow. This is the longest phase and it is called as the first growth phase or G1 phase. After this, if the cell decides to replicate, it will replicate its DNA. That replication of DNA, that phase is called synthetic phase or S phase. After that, there is a short growth phase which is called G2 phase. What happens during this time? This time, the cell synthesizes a few more organelles and certain proteins needed for cell division. It is like this: your mother is planning to prepare kheer, and she has got milk, sugar, rice and other things to prepare the kheer. And when she starts preparing, just before she starts preparing, she discovers that there is no cardamom. So she will have to buy cardamom so that the dish can be prepared. Last minute preparation, that is G2 phase. It's a short one. After that, the cell goes into cell division. That is called phase of division, cell division, or dividing phase, or mitotic phase. So now let us go to the muscle cells, the heart muscle cells, and the neurons in our body. These are two examples of cells in our body which after origin just grow, develop, differentiate, and start on doing their function and never divide. Opposite is the example of the skin cells. The function of skin cells makes it vulnerable to breakage. So they have a high cell division rate and their cell cycle is almost 24 hours. That is, the cell cycle will be repeated after every 24 hours. Now, why do the neurons or the heart muscle cells do not divide? They do certain vital functions. These functions cannot stop even for a second. So, if they undergo cell cycle, then the connectivity between different neurons, the supply of blood to different parts of the tissue will be temporarily stopped. And this is not possible for the survival of the organism. So, these tissues are designed by environment. Just imagine the engineer in the nature which has designed these cells in such a way that they can stay without dividing throughout their life. Dear students, imagine that on the day of your examination or a day before your examination, your heart muscle cells tell you, I want to undergo cell division. I'm off for 24 hours. You mind your body yourself. Just imagine what will be our plight. So, that is the reason why those cells, like the heart cells and the neurons, do not divide. So coming to the cell cycle, there are three stages which are together called as interphase. Which are those three stages? Stages G1, S and G2. Now what happens in these stages? In G1, the cell grows, accumulates nutrients for growth, uses it, stores some, and synthesizes amino acids and proteins necessary for the functioning of the cell. Then in the S phase, now the cell has decided to divide. So it moves on to the S phase. It replicates its DNA. The whole replication of DNA happens in the S phase. After replication, it will form new organelles and new proteins just needed for cell division. That is a very short phase, which is G2 phase, the second growth phase. All these three phases together, G1, S and G2 are together called as interphase. Now the whole preparation for cell division is done. DNA has replicated, growth has happened, material needed for cell division has been synthesized. Cell moves on to the next stage, which is called the dividing phase, which is also called the mitotic phase. M phase or mitotic phase, what happens here? Here the actual cell division takes place. Now this cell division also takes place in two major steps. The division of the nucleus, which is called as karyokinesis, followed by the division of cytoplasm, which is called cytokinesis. Now these two will be dealt in detail in the next module. Dear children, cell teaches us a great lesson. Nothing is good or bad. When it happens in our life, whether it is good or bad, that decides it. How? Cell has to divide for growth. Cell has to divide for reproduction. Cell division is very, very essential. But when a cell divides unnecessarily, unwantedly, then it ends up in cancer. What is cancer? Cancer is unlimited division of cells when not needed. How does it happen? There are actually three checkpoints during the cell division. One is at the G1 stage, second is between the G2 and M stage, and third is at the spindle formation stage. Now these you will be learning in higher classes. Now if these checkpoints don't work properly, it is like the brakes in a car not working, then the cell continues to divide without any reason. That ends up in cancer. So now let us learn a very important concept related to cell, what you call by the term karyotype. If I take any organism and I take one cell of that organism, I pick up all the chromosomes in it and arrange it in a particular organized manner. That is, I arrange them with the shorter arms on top and the longer arms on the bottom in a systematic way. Then whatever I get is called a karyotype. So what is a karyotype? It refers to all the chromosomes in any cell arranged in a very particular manner. Now here the diagram shows the karyotype of a human being. It shows you that we have 23 pairs of chromosomes. The first 22 pairs, they look like pairs because both the chromosomes in a pair are similar in size, shape as well as structure. How do you know they are similar in structure? You can see the banding in them. The lighter banding is because of the DNA which is loosely coiled, and that is called euchromatin, and the darker bands are due to heterochromatin, which are tightly condensed and coiled DNA. So the banding is also similar, so the structure is also similar. These 22 pairs of chromosomes, which normally control the body character, the somatic character of an organism, are called autosomes. Look at the 23rd pair, which you can see in a red square. The pair, though we call it a pair, look dissimilar. One is longer, bigger, the other one is shorter. This is a pair of sex chromosomes or allosomes in human body. Now, the longer one is called X chromosome and the shorter one is called a Y chromosome. As this particular cell has one X and one Y, this cell belongs to the body of a man. So, in human body, 22 pairs of chromosomes are autosomes which control the somatic or body characters, and the 23rd pair is allosome or sex chromosomes which control sexual characters. Apart from that, they control a few other characters which you'll be learning in the upcoming modules. Now, if you take the cell of a sperm or an egg from human body, and you examine the karyotype, then you will realize that the chromosomes are not found in pairs, the chromosomes are found in single set. Then the question arises in your mind, if the testis of a man or the ovary of a female has got chromosomes in pairs, how come the sperm or the egg has got the chromosomes not in pairs? Now, this unpaired set of chromosomes in any cell, that condition is called as a haploid condition, or we can reword it. A haploid cell is a cell where the chromosomes are found in single sets, and diploid is that condition where the cells have chromosomes in pairs. Typical example, the cell of an ovary and testis is diploid. The cell of the gametes, that is the sperm and the egg, are haploid. Now, how does a haploid cell arise from a diploid organ? Now, that is a question which will be answered when we learn about the different types of cell division. Let us now proceed to the question answer session. Distinguish between the following pairs: haploid and diploid according to the number of chromosomes. Haploid has got single set, whereas diploid has got chromosomes in pair. Autosomes and allosomes according to the characters controlled. Autosomes control body character like eye color, height. Allosome control sex-linked character and also certain characters like color blindness and hemophilia. Karyotype and karyokinesis. Karyotype definition is the number and type of chromosomes in a cell, and karyokinesis is the division of nucleus during cell division. Moving on to the diagram-based questions. There are 2 diagram-based questions given in this lesson. So let us go through that. The first one says, how many pairs of chromosomes are represented here? Give a suitable heading. How is the pair represented inside the red square different from that represented in the green square? And Seema is confused whether the diagram belongs to a skin cell or a sperm cell. So, how many pairs? 23 pairs. Heading, human karyotype. Red one is autosomes, and in green square it is allosomes. And as the chromosomes are in pairs, the cell is a diploid cell. So it can only belong to the human skin cell. Diagram based question. Label 1, 2 and 3. 1 is G1, 2 is S and 3 is G2. How is 1 different from 2? In 1 it is growth phase, synthesis of proteins and amino acids happen. 2 is synthetic phase where DNA replicates. What does the letter M denote? It denotes the dividing phase or the mitotic phase. Why is stage 3 so short? The stage is short because it only makes the material needed for DNA repair or spindle formation. So dear children, we have come to the end of module number 4 under genetics. In the upcoming modules, we will be learning about different types of cell division, both mitosis as well as meiosis. Think science. Keep learning, keep watching. Have a nice day and bye.